As I said, we are going to continue with the presentation, and now I deal with Alan Pape and GSR.
Good afternoon, everyone. So, as Sander just said, my name is Alan Pape, and I'm going to talk to you about the status of the meiofauna baseline surveying in the GSR claim area. So, although it was already mentioned by the general manager of GSR, Chris van Neye, yesterday, I will just briefly explain again who GSR is. So, GSR is a private Belgian company, and the letters stand for Global Sea Mineral Resources. And in 2013, they signed a contract with the International Seabed Authority for the exploration for polymetallic nodules. So here you see a map of the different claims in the Clarion-Clipperton fracture zone in the northeastern Pacific. And here is the location of GSR. So here is GSR, and it's located in the eastern parts of the Clarion-Clipperton fracture zone. And you can also see, I hope you can see, that it's also divided into 3 zones. So you have the darker green areas, those are the 3 areas of the GSR claim. I will also briefly recapitulate about the Marine Biology Research Group, which is the research group I'm part of. It's a research group at Ghent University. It was founded in the 1970s. Its main expertise is the meiofauna, and most meiofauna research is actually revolved around nematodes. It's a fairly large group. You can see here a group picture of the lab. But there are also some people that are involved in deep sea research projects, but there are also other research projects going on about the Antarctic, about mangroves, about estuaries. So we have a great diversity of research projects. And last but not least, the last 2 years we have been involved in a service contract with GSR for the environmental baseline study in their claim in the Clarion-Clipperton fracture zone. So GSR signed the contract in 2013, and the first sampling campaign took place in 2014, so that was last year. Well, in terms of biology, it was actually a pilot study. Why was that? Because no replicates were taken. So we didn't define a number of stations, and we took several replicate samplings there. Also, to study the benthos, the box scorer was taken. So this is the box scorer, this is a picture of a box scorer. This is not the most suitable sampling device to study the meiofauna because it generates a large bow wave. So you get the fluffy upper sediment layers that are being blown away. And this was actually also proven in our samples from this campaign, because we found actually quite a lot of nematodes in the water that is standing above the sediment in the box scorer. So we also sampled that, and then we looked at that, and that's how we saw this. So this is a map actually showing the 3 zones of the GSR claim, and the red dots represent the different box scores that were taken. So in total we took 10 box scores, and they were distributed over the 3 zones. So how did we subsample the box score? Because we did subsample the box score. In each box score we took 3 cores at random for meiofauna analysis. Then we collected another core to look at environmental variables such as total organic matter content, total nitrogen content, grain size analysis, and also another two cores were selected by the geologists. And then half of each box score was intended for macrofauna. So actually we got a rather limited set of samples, but we tried to maximize the amount of biological information from these samples to get an idea about the benthic ecosystem over there. So the meiofauna cores were sliced, so they were cut into different layers. We cut them per 1 centimeter layers down to 5 centimeters. So because we took subcores in a box core, this was actually possible. There were no nodules, or not very big nodules inside the core, so slicing was possible here. So what are the results of these 2014 samples? I have here a graph. On the x-axis I have total megafauna abundances, expressed as number of individuals per 10 square centimeters. Now 10 square centimeters is the general area unit that is used to express megafaunal densities. And here on the y-axis, these are different studies that have been published on megafauna in nodule areas. And on the first line we have the results of our study. So let me first explain also what these dots mean. So if I have two dots, it's the minimum and the maximum densities, and then if I have one in the middle, it's the average, and if I have only one, it's the average megafaunal densities, as reported in the papers of these studies. So what we actually saw was in our study, densities range between 43 and 108 individuals per 10 square centimeters, and this is actually more or less in the same range as the other studies conducted in other nodule areas, including those in the CCFC. So, as I mentioned, we also sliced the sediment cores, so we could have a look at the vertical distribution of the megafauna in the sediment. And what we saw was that densities decline with sediment depth, which is actually commonly seen in marine sediments, including deep sea sediments. We also took a look at the megafaunal taxon composition. So here on the horizontal axis you have the different box scores that were taken. On the y-axis you have relative abundance. And what you can immediately see is that there is one particular taxon which is very abundant. I have to say this is metazoan megafauna, so we didn't look at the protozoan megafauna. And what we can see is that nematodes rule. That's the main conclusion. Other taxa that are also quite abundant, that are the second most abundant in our samples, are the copepods, Pedro's favorite animals, and then also the nautri larvae. What is also interesting to notify is that there was one box score, namely box score number 5. This one did not contain any nodules, but we did not see any real difference in higher taxon composition compared to the box scores that did have nodules. Of course, this is only one box score. We cannot make any real conclusions about this, no statistically found conclusion, but it's interesting to note. The same is true for total densities of megafauna. We also found a lot of nematodes in our macrofauna samples. So the macrofauna-sized nematodes, we call them. They are bigger, so they are retained on a sieve of 250 microns. We hand-picked them, and we also mounted them on slides for light microscopy. And we started to identify them, but it's still a process that is in progress, actually. What we can say is that many of these macrofauna-sized nematodes are belonging to the order of the inoplids. So what I found a lot were these nematodes with these large teeth. I don't know if that's very visible. Also, what I try to do is take pictures with the microscope camera, and also I try to make sketches of taxonomically important features, which can serve as a basis of a kind of nematode atlas for the GSR claim. So it can be consulted later on. So this year we went on our second sampling campaign. And there we took, now we took a different approach. So we actually visited only 2 zones this year, B4 and B6, and we concentrated on 3 different stations, which are located here, and they are indicated here by the red rectangles. And within each station, we took replicate multi-core deployments. So we took a multi-core now, in addition to a box core. The multi-core was used to sample the megafauna, and it's a much more suitable sampling device for the megafauna because it produces relatively undisturbed sediment core, so it's much better than the box core. Also interesting to note is that the 3 replicate deployments within each station were about 1 kilometer's distance from each other. Also an interesting fact is that the station located in zone P6 overlapped with the station that was sampled during the JPI Oceans campaign. So the GSR campaign took place in September, October 2015, so it's very recent. And the JPI Oceans campaign took place in March, April 2015. Now what we did was, we chose 3 multi-core locations, so the 3 deployments were in the exact same position as 3 of the 5 MUC locations of the JPI Oceans campaign. So this enables us to look at the intra-annual variability in meiofauna communities, which is actually one of the recommendations made by ISA. So this is a picture of the MUC core. So the multi-core that we used had 8 core liners. 3 of them were finally used for meiofauna community studies. So what we did is we took the top 5 centimeters of each core, and we did not slice them because nearly all of the cores contained big nodules, which was very annoying. We really hated that, so we couldn't slice it. So I would love to hear more about the special technique that Andy has to slice cores with nodules, because I'm very curious about that. So one of the cores we preserved for the study of the morphology of the meiofauna. So this core was stored in formaldehyde. Then another core, we stored the 0 to 5 centimeter layers in DES. So this is the solution containing dimethyl sulfoxide and EDTA, saturated with sodium chloride. So this one was intended to voucher and barcode individually the nematodes inside the sediment. Then a last core we stored at -20 degrees, because at first we knew we wanted to do some molecular analysis with these sediments, but we didn't know in the beginning what we were going to do. So then afterwards we decided to extract the meiofauna, and then the extract was actually already sent to a sequencing company for next generation sequencing. So for these samples, I do not have any results yet, so I cannot really say a lot about them yet. I cannot tell you if they have been successful or not. But for the other 2 cores, I have some very preliminary results. Very preliminary. So for the morphological study of the meiofauna, I have looked at 3 samples already. And I've looked at the taxon composition, and what is immediately, again, very clear is that there is one taxon, very abundant, which are the nematodes, again. So, the nematodes rule. And again, the same thing, copepods and nautri are the second most abundant groups of the metazoan meiofauna. So the samples stored in DES we used to voucher and barcode the nematodes individually. I will first show you how we did it in practice. So each sample stored in DES, we actually first extracted the meiofauna by using the redox centrifugation technique. The extract was again preserved in the DES solution, and then we picked nematodes individually from this sample using a stereo microscope and a fine needle. Then we transferred one nematode at a time. Actually, always one nematode. We transferred the one nematode to a drop of autoclaved milky water, so sterile water, on a slide. We closed the slide again with a glass cover slip, and then we looked at it through a microscope. Then I identified this nematode to the lowest taxonomical level possible, which is generally genus, and I vouchered it. So I took pictures of different taxonomically important characteristics. And then I try to open, always very carefully, because this is a very delicate process, open the slide again, try to find the nematode again, because that can be very difficult and very frustrating. And then I transferred the nematode to an Eppendorf file containing a drop of worm lysis buffer. This Eppendorf file is unfrozen at -20 degrees, where it will stay until the DNA is extracted. And then we amplified the COX1 or CO1 and the 18S genes. And the purpose is that we can get sequences for these 2 markers, and that we eventually wind up with a reference database of sequences. Now, why do we want this, you can ask? Well, this can help us to determine connectivity within areas and also between areas. So if we find a certain genus in one station of the GSR claim and we also find in another station, is it the same species? But we can also compare between the GSR claim and the BGR claim, for instance, which can tell us a lot about connectivity between different claims. So what are the first results? So far I have vouchered 90 different nematodes. These 90 nematodes belong to 36 different genera. So actually here I have the list of genus names, but it's quite small, so I don't know if you can read it. The most frequently vouchered nematodes were belonging to genera like Acantholimus, Viscosia, and Hellimus. The first 30 specimens were already subjected to DNA extraction and PCR reactions, and what is interesting is that 50% of these nematodes had either one of the two markers amplified or both. So there was not a very high success rate. So 30% of the POX1 and about 30% of the 18S markers were actually amplified, but there was no correlation between the two. So it could be that one nematode had a CO1 marker amplified, but not the 18S. And so this is kind of, but these are very preliminary results, so we still have to look at it further. So what are the plans for next year besides and this year, to finish the analysis, of course. But in terms of sampling also, I was thinking about what we could do. We can sample the same stations to have a time series. We can also, of course, it would be wise to sample more stations to have a wider spatial spread of stations and more data to increase our database of DNA sequences and morphological data. And also it would be interesting because now we have only sampled stations with nodules. Maybe it would be interesting to compare with stations containing no nodules if these can be found. Of course, it would be interesting to increase replication. Instead of 3, take 5 replicates, because the more replicates, the higher your power, your statistical power. It would also be interesting to collaborate with other contractors in terms of biological data, then we can have a real view of connectivity throughout the area. Also sample the areas of particular environmental interests, which would be very nice. There's one close to the GSR claim. And also, during our last campaign, we saw several dredge tracks in the claim area. It would be very nice if we could visit them with an ROV and conduct some targeted sampling with push cores inside the disturbed areas and outside, so we can investigate the recovery of the communities. So that was it. Thank you for your attention.
Some questions?
So I'm a little bit surprised that you didn't find any tubularians in your sample.
Yeah.
Because it seems to be very abundant.
Did you find tubularians?
No. So therefore, I'm surprised that you also didn't find any. Do you have any comment on that? I don't know. We never found some.
It's the
Yeah,
Ludo.
it's the
What happened to the Ludos?
They explode. There's a fresh water in the Ludo.
You need special extraction techniques for the blood.
That's why we
we
found
are
them.
we are not using Ludos, we are using Levas.
Yeah, but that's the same as Ludo. Yeah, fresh water.
Oh, I don't know. I don't know.
When you said that you do NGS on extracted meiofauna?
Yeah.
What does it mean, extracted meiofauna? It's after Ludo?
Yeah.
Yeah, indeed. After the Ludic centrifugation,
Ah,
we keep the supernatant, and then this is further used for the NGS.
did you already get any results of it? Or?
I don't think so.
So they were sent to a company,
Okay.
and so they're being processed now. So we hope to get the data after Christmas.
Okay.
To get many forerunners in the Ludox. I'm interested to know whether Ludox extracts forerunners.
I have to say I didn't really pay any attention, but I didn't see any colored rose Bengal stain, because we did stain the samples with rose Bengal. and we didn't see any stained forearms in the samples.
Yeah, Mr. But how do you voucher? Do you take pictures or do you make drawings?
Pictures.
Pictures.
Yeah.
No videos.
No, we don't have that capability yet.
Just referring to the fact that you use a multi-core, which I now know what it is, and you've got one core with formalin and one with DES and one with probably DES and NGS. You must know how similar those different cores are to each other. I'm just thinking about, is it at all possible that you could be sampling subtly different environments, even within the radius of the actual core? Well, presumably you must have studied this until the cows come home with morphology.
What do you mean, how different they are?
Well, I've often, back in the day, I've often thought about this. If you were sampling one core for morphology or barcodes and then another core for NGS, it could be that if this community was just subtly different due to, microscopic characteristics of the sediments, that it wouldn't, these guys might be different to those guys. Does that not happen?
Yeah.
Yes, of course
Jana?
it happens. I mean, you have heterogeneity only due to the presence of the nodules. For instance, one core could have a nodule and the other one you're using for NGS doesn't have a nodule. But the idea of the barcoding is to get a reference database so we can link our barcodes with what we find in the NGS. So that's the idea of the.
And also we took replicate deployments.
Yeah.
So we took several MUC cores in one station. So.
I think there's the problem is what I show in one of the papers show that there's not very much of overlap if you take different cores in terms of sequences. So of course if you do eDNA and you benchmark on other core, that may not be really good calibration. You should really work with the same segment.
Like you were saying, one thing that could be done potentially is taking a core and then homogenize it and then
this is the
take a subsample of the community DNA potentially.
you should do it like that because otherwise there's no calibration. The
No.
cores can be different.
Yeah, but okay, that is why you replicate. You take your 3 replicates for NGS. I mean, the barcoding approach is not meant to characterize your community. The barcoding approach is
It
only
depends what
meant.
you look at, but if you do NGS on eDNA, this is just you have to combine them or mix them because it's not just to solve the specimen barcode, it's just to get everything out of this.
No, but we are not doing eDNA. We're doing NGS on the myofana community.
But the barcoding process should happen on a subsample of the community that goes into the NGS, and then you've got a higher possibility, a higher chance of being able to track those guys in your NGS data set.
Yeah, but actually the idea is to create such a big data, I mean to voucher so many nematodes that at some point you have a reference database and that you can go back to that one. But my concern is by homogenizing and then splitting up, it increases the treatment of your sample.
Well, maybe I just add to your comment here. So with the 3 replicates, you describe the population rather than a sample. That's what you get. So it's much more reliable than a single sample.
It depends what your goal is. I mean, if the goal is to see if there's a good fit between morphology and the community DNA, then you do homogenizing. But if what you're doing there is perfectly fine, it's such a different focus.
I don't know if you could show that one of the early slides about the abundance in your area compared to the others.
This one.
Yeah. And I don't know if what struck me today is that there's quite a lot of variability in the total abundance of myofauna and nematodes, therefore, between different presentations that I've seen. And maybe even more than is apparent here. And I'm wondering why that is.
Well, there are several reasons for that. First of all, some studies use different sieve sieves as a lower sieve size limit. So we use 32 micron, which is the universally accepted lower sieve size for megafauna. Some of them used 63 or even higher, so that can make a very big difference, I think. And also the sediment depth interval also slightly varied between these studies.
Right, and also presumably there's, depending on if you're on the east of the CCZ
Yeah.
or the west
There's
or
also
the middle,
difference, yeah.
there'll be differences. And so what I was wondering for this workshop was, would it be useful if somebody could take the data that we are being presented with, maybe tomorrow, and sit down and just plot or write down all the abundances that we've been seeing, and maybe try to make some sense of it.
That would be very interesting.
It would
Yeah.
it'd be useful if someone could just look at that, because unless, well, you've already got me a job for tomorrow, and it's better if someone with my fauna with sense, not me, actually did this, because it could also be problems with how people deal with their samples, nothing to do with the environment, that we're getting these differences. It may be environmental, it may be something simple, it may be something more complex. And I just think it would be useful while everyone's here and with all these data to just maybe have a first look and try and see whether there's something that might guide us in this workshop in our recommendations.
Yeah, that's a very good suggestion.
Thank you very much.
Okay, thank you.